Have you ever wondered how life begins at cellular level? It all starts with gametogenesis, the process by which our body produces gametes, sperm in males and egg in females. In this video, we'll break down the entire process in simple and clear way. So let's get started. What is gametogenesis? The process of formation of male and female gametes in testes and ovaries respectively is called gametogenesis. It is of two types, spermatogenesis and oogenesis. This process occurs in gonads, testes in males and ovaries in females. It involves cell division, process like mitosis and meiosis, ensuring gametes have half the number. Let's start with spermatogenesis. Spermatogenesis is the process of sperm formation occurring in testes. It takes place in seminiferous tubules of testes. It occurs continuously after puberty, and it follows different stages like spermatogonia undergoes mitosis and forms primary spermatocytes. Primary spermatocyte undergoes meiotic division, means meiosis, and forms secondary spermatocytes. Secondary spermatocytes undergo second meiosis and form spermatids. Spermatids forms spermatozoa by differentiation, and this method of differentiation is called spermiogenesis. And spermatogonia, it is immature cell that undergo meiotic, means mitosis division, and increases its number. These cells enter primary spermatocytes enters into first meiotic division and forms secondary spermatocytes. Spermatogonia and primary spermatocytes are diploid, means they have a 46 number of chromosomes per cell. Primary spermatocytes undergo first meiotic division, means reductional division, and forms secondary spermatocytes. Each primary spermatocytes, means one primary spermatocytes form two secondary spermatocytes, form two haploid secondary spermatocytes. After meiosis 2, secondary spermatocytes form spermatids. Secondary spermatocytes form spermatids, and these spermatids finally forms spermatozoa by differentiation method, and this differentiation method is called spermiogenesis. Spermatids and secondary spermatocytes have 23 number of chromosomes. It means these both are haploid. This entire process of spermatogenesis takes about 64 days. ये जो पूरा प्रोसेस है स्पर्मेटोजेनेसिस का, ये 64 डेज में कंप्लीट होता है और मिलियंस ऑफ स्पर्म The head of sperm remains embedded in Sertoli cells and are released from seminiferous tubules by the process of spermiation. Spermiation and spermatogenesis. Spermiogenesis, these are two key words. Spermiation is the process of release of sperm head from the Sertoli cells inside the seminiferous tubules, and spermiogenesis is the formation of spermatids to spermatozoa. These are the two key words, spermiation and spermiogenesis. Hormone control of spermatogenesis. The regulation of spermatogenesis is controlled by main three hormones. First, gonadotropin releasing hormone, follicular stimulating hormone, and luteinizing hormone. These three are the main hormones. Gonadotropin releasing hormone, GnRH, secreted by hypothalamus. Hypothalamus secretes GnRH, gonadotropin releasing hormone. This gonadotropin releasing hormone stimulates the anterior pituitary to release two main hormones, that is FSH and LH. Follicular stimulating hormone, FSH, follicular stimulating hormone, stimulates the Sertoli cells in testes to support and nourish development of sperms. Sertoli cells को stimulate करता है for spermatogenesis, means support and development of sperm, and Sertoli cells release another hormone called inhibin, which help in regulation of sperm formation. Anterior pituitary release another hormone, LH, which is luteinizing hormone, which stimulates Leydig cells which is present in testes, to secrete hormone testosterone. Testosterone, the key hormone for sperm production, means spermatogenesis के लिए. Testosterone maintain male reproductive structure and secondary sexual characteristics. The two hormones, inhibin and testosterone, these are beneficial. They support in spermatogenesis, but when they are in very high amount, they give negative feedback and prevent the overproduction of sperm. The inhibin, which is secreted by Sertoli cells, give negative feedback to anterior pituitary and stop the production of FSH and prevents overproduction of sperm. And the second hormone, testosterone, which is secreted by LH, it gives negative feedback to anterior pituitary and hypothalamus and stops the production of hormones and prevents. Body maintains balance in sperm production using a negative feedback loop. जो हमारी बॉडी है वो ओवर प्रोडक्शन या लेस प्रोडक्शन ऑफ स्पर्म से प्रिवेंट करने के लिए, मीन्स ओवर प्रोडक्शन ऑफ स्पर्म से प्रिवेंट करने के लिए एक नेगेटिव फीडबैक लूप बनाती है. अगर if sperm production is too low, the hypothalamus releases more GnRH stimulating hormone, GnRH stimulating FSH and LH secretion, which increases testosterone and sperm production. If sperm count is too low, inhibin is released to suppress the release of to suppress FSH production, reducing sperm formation. Similarly, high level of testosterone inhibits GnRH and LH secretion, maintaining hormonal balance and prevents the overproduction of sperm. Now let's start oogenesis. Oogenesis is the process of formation of mature female gametes. It is started during embryonic development. It takes place in ovaries. Unlike spermatogenesis, this process begins before birth and pauses at different stages. It follows steps like oogonium, form primary oocytes and secondary oocyte, first polar body, and forms a ovum and second polar body. The process of formation of mature female gamete is called oogenesis. It started during embryonic development stage, when millions of oogonia are formed in each fetal ovary. Oogonia start division and enters into prophase 1 of meiotic division 1. जो oogonia है, oogonia start division. These are female germ cells. Oogonia are the female germ cells that undergo mitosis to increase in number. Form primary oocyte. By undergoing mitosis, it forms primary oocyte. Primary oocytes get arrested when it enters meiosis 1 at the prophase 1 before birth. Primary oocytes जो कि present होते हैं at birth, arrested in the prophase 1 of meiosis 1. Then at puberty, one primary oocyte completes meiosis 1 and जो एक primary oocytes है at puberty, after puberty जो एक primary oocytes है वो एक meiosis complete करता है and produces a large secondary oocytes and one first polar body. This secondary oocyte is again arrested at the metaphase phase of meiosis 2. At puberty, one primary oocyte complete meiosis each month, producing a large secondary oocytes and a smaller polar body. The secondary oocyte entered meiosis 2 but pauses metaphase 2 until fertilization. जो primary oocytes है वो आफ्टर प्यूबर्टी सेकेंडरी ओसाइट बनाती है, जो कि लार्जर सेकेंडरी ओसाइट होती है, और एक फर्स्ट पोलर बॉडी बनाती है. जो लार्जर सेकेंडरी ओसाइट्स है वो फिर अरेस्ट हो जाता है. पहले वो प्राइमरी ओसाइट अरेस्ट होता है बिफोर बर्थ, और उसके बाद सेकेंडरी ओसाइट है वो अरेस्ट हो जाता है एट मेटाफेज 2. इफ फर्टिलाइजेशन ऑकर्स, अगर फर्टिलाइजेशन ऑकर होता है तो जो मियोसिस 2 है वो कंप्लीट होता है फॉर्मिंग अ मैच्योर ओवम. मैच्योर ओवम फॉर्म करता है और एक सेकेंडरी पोलर बॉडी फॉर्म करता है. देखो, फर्स्ट अरेस्ट होता है प्राइमरी ओसाइट जो कि बिफोर बर्थ एम्ब्रियोनिक स्टेज में होता है. फिर सेकेंड जो है सेकेंडरी ओसाइट अरेस्ट होता है मेटाफेज में मियोसिस 2 के, जो कि अरेस्ट जो हुआ है वो रिलीज होता है जब वो फर्टिलाइजेशन होता है एग का. मतलब सेकेंडरी ओसाइट जब फर्टिलाइज होती है तब वो ओवम फॉर्म करता है और एक सेकेंडरी पोलर बॉडी फॉर्म करता है. तब पूरी की पूरी oogenesis कंप्लीट होती है. Oogonia start division and enters into prophase 1 of meiotic division and get temporarily arrested at the stage called primary oocytes. Primary oocytes get arrested at prophase 1 of meiosis 1. Each primary oocytes surrounded by a layer of granulosa cells, then it is called primary follicles. जो प्राइमरी oocytes है वो ग्रेनुलोसा सेल्स, एक जो सेल होता है उससे सराउंडेड होते हैं और फिर हम उसको प्राइमरी फॉलिकल्स बोलते हैं. At puberty, about 60,000 to 80,000 of primary follicles are present in each ovary. फीमेल्स में 2 ओवरी होती हैं, एक लेफ्ट एक राइट. हर एक ओवरी में एट प्यूबर्टी, जब प्यूबर्टी हिट करती है तब 6 primary follicles get surrounded by more layer of granulosa cells. जब oocytes एक नॉर्मल ग्रेनुलोसा सेल से सराउंड होते हैं तो हम उसको प्राइमरी फॉलिकल्स बोलते हैं. बट व्हेन primary follicles get surrounded by more layer of granulosa cells are called secondary follicles that transform into tertiary follicles that contains a liquid called antrum, fluid cavity called antrum. फर्स्ट प्राइमरी फॉलिकल्स फॉर्म होता है, फिर वो और लेयर्स ऑफ ग्रेनुलोसा सेल से सराउंड होता है, फिर वो सेकेंडरी फॉलिकल बनाता है. देन वो जो सेकेंडरी फॉलिकल है वो ट्रांसफॉर्म होकर टर्शरी फॉलिकल बनाता है. टर्शरी फॉलिकल का स्पेशलिटी है कि उसमें एक एंट्रम प्रेजेंट होता है, मीन्स फ्लूइड कैविटी प्रेजेंट होता है जिसको हम एंट्रम बोलते हैं. The tertiary follicle further changes into mature follicles called Graafian follicles. जो tertiary follicles होती है वो further change होती है into mature follicles called Graafian follicles, which ruptures to release secondary oocytes, means ovum को release करता है Graafian follicles, form the ovary by the process of ovulation. Should see this flow chart. Like oogonium undergo mitosis from primary oocytes arrest at prophase 1, and then primary oocytes go meiosis continues after puberty. This is before birth. Oogonium undergo primary mitosis and from primary oocytes, and after birth, after puberty, जो arrested रहता है वो after puberty primary oocytes goes meiosis continuous and forms secondary oocyte and one smaller polar body. Then this secondary oocyte get arrest again in metaphase 2, which is released when sperm comes and fertilizes the ovum, and the secondary oocytes form ovum and first secondary polar body, and this oogonia is completed, means oogenesis is completed by this after fertilization. The key difference between spermatogenesis and oogenesis. Spermatogenesis, oogenesis. Spermatogenesis occurs in testes and oogenesis occurs in ovaries. Spermatogenesis form male gametes called sperm. Oogenesis form female gametes called ovum or egg. 4 sperms are produced from each spermatogonium, and in oogenesis only one functional ovum is produced from each oogonium. Spermatogenesis maturation division is equal without polar bodies, and in oogenesis maturation division is unequal with larger ovum and 2 smaller polar body. It's not 3, it's 2 smaller polar body The sperm are smaller in size, greater in number, and motile. In oogenesis, the eggs are larger in size, fewer in number, and unlike male, females produces only one egg per menstrual cycle, and the total number of eggs is predetermined at the birth. Here, this two flowchart is given of spermatogenesis and oogenesis. This flowcharts clearly explain the process of spermatogenesis and oogenesis. Spermatogenesis, formation of male germ cell, and oogenesis is the formation of female germ cells. Spermatogonia forms primary oocytes, and primary oocytes form secondary oocytes. Secondary oocytes form spermatids, and each spermatid forms sperm. And in oogenesis, oogonium undergo mitosis from primary oocyte. Primary oocyte get arrested at prophase 1 of meiosis 1 before birth, and after puberty, meiosis 1 is completed and the secondary oocyte is formed, and it get arrested at metaphase 2 of meiosis 2 and forms first polar body. When fertilization occurs, an ovum is formed and second polar body is formed. This is the complete information of gametogenesis. If you found this video helpful, don't forget to like, subscribe and share for more biological lessons. Comment below if you have any question. See you in the next video.